Hello, and welcome back to another episode of Lab Talk, the series where we share expert tips and best practices to help you get the most out of the products in your lab. Today is Thursday, May 16th, and we have a special treat for you guys today. Our topic is all about minimizing contamination in CO2 incubators. And what you may or may not know is that Thermo Fisher Scientific has been in the CO2 incubator space for a long time, since 1968 to be exact. And over the years, we've heard your questions, and our specialists have been responding to those questions with their wealth of expertise. And today, we're lucky to have two of those CO2 experts with us. And I'll be asking them some of the hottest questions when it comes to cell culture contamination. So it's my pleasure to welcome Mary Kay Bates, Senior Global Application Scientist with Thermo Fisher Scientific. Mary Kay has a background in cancer biology and gene therapy research and development. Also joining us today is Callie Heisel, Commercial Manager with Thermo Fisher Scientific, who has over 20 years experience in the CO2 incubator market. Welcome to you both.
Thanks, Megan. It's great to be back at Lab Talk.
Yeah, thanks for having me, Megan.
It's great to have you both. All right, so today's Lab Talk will be a little different than our previous episodes. I mentioned we have a special treat for you. Today's episode will be more in the style of a talk show rather than a presentation. So it's a contamination chat, if you will. We'll have some visuals up in the presentation window, but the majority of our time today will be spent discussing the most commonly asked questions, around controlling contamination in CO2 incubators. Let's kick this off with our first topic of conversation. Mary Kay, let's start with you. So we know cell culture contamination is a continuing challenge. Where does contamination even come from?
That's a great question. And a lot of people don't know this. In a standard lab, there are two main sources of contamination. The first is from us, the people in the lab, and the second is from the indoor air around us. As humans, we all carry 10,000 microorganisms per square centimeter on our skin, and normal indoor room air contains 30 to 1,000 microorganisms per liter of air. So these numbers are based on published peer-reviewed studies, and I think it kind of helps explain where contamination comes from.
Oh, I didn't realize so many microorganisms lived on my skin. I kind of feel like I need to go take a shower. That actually kind of leads me to my next question. If microorganisms live everywhere, then it seems nearly impossible to keep them out of cultures and out of the incubator. So are there some best practices you can share around that?
Yeah, and I can take this one, Megan. As home for cultured cells, the CO2 incubator is arguably the most important piece of equipment in the lab. If your cells don't grow properly, or if they get contaminated, you won't have anything on which to experiment. But every time you open the incubator door, and that's any incubator, contamination can enter. The best practice is to clean the incubator on a regular basis, try to keep the incubator door closed as much as possible, and ultimately use an incubator that is designed to help protect your cells.
Okay, so, I'm hearing proper cleaning, one, and design, two, are both important, but those are pretty broad statements. Can we break that down a little further? Let's start with some cleaning methods and then move into design. Can you share some best practices for cleaning a CO2 incubator?
Yeah, I'll jump in here. We all know that cleaning a CO2 incubator is not the most fun job. So it's good to have a model of CO2 incubator that is easy to clean with smooth chamber walls in the inside where the shelving components are easily removed, and you can put them back in easily too.
Exactly, Mary Kay. Most incubator manufacturers recommend cleaning with a mild dish soap and water, a clear water rinse, followed with 70% ethanol and letting that air dry. The ethanol is not only a good disinfectant, but will also remove any residues that could build up over time.
Thanks, Callie. That sounds easy enough. I think I could clean these incubators behind me with doing that. Are there any interior treatments that could make it any easier to clean?
And absolutely. A great example are the different finishes of stainless steel that we see in CO2 incubators. Essentially, the smoother the stainless steel, the easier it is to clean. For context, some CO2 incubators are finished with brushed stainless steel, or some are polished. Brushed stainless steel is a lower quality because the brushing adds small grooves where microorganisms can hide, and this can be more challenging to keep clean. Mechanically polished stainless steel provides a smooth and polished surface, and it's easier to clean than brushed stainless steel. And best of all is electropolished stainless steel. This treatment removes microscopic structure and, according to published reports, can reduce areas where bacteria can attach by up to 90%. And Megan, we definitely cannot forget about copper, which is naturally an antimicrobial.
Kelly, I've heard about copper. One of my incubators behind me is actually copper, but I don't really understand that how that works. Can you explain that a little bit?
Ooh, I get to talk about the copper. For thousands of years, copper has been used to inhibit infection. The ancient Greeks and Aztecs used copper as a topical treatment for skin wounds. Much more recently than that, though, in 2006, the US EPA certified copper as the first solid antimicrobial after testing more than 300 alloys. And the EPA determined that only alloys with more than 60% copper content had any antimicrobial activity, and that the effectiveness of the copper is proportional to the copper content. So that means that 100% pure copper is the most effective. Now, the exact mechanism of killing is still being researched, but it appears that oxidized copper breaks down disulfide bonds in proteins, and once in contact with nucleic acid, the copper ions take only seconds to destroy that structure. There are hundreds of publications showing that solid copper can eliminate all sorts of viruses, bacteria, and fungi. It's really interesting reading if you want to dig into that. But I also like to mention copper is the world's most recycled material.
Hmm. That sounds pretty incredible. But, you shared some great benefits, and I'm all about that. But I got to ask, are there any downsides to a copper interior for an incubator?
Well, in general, copper is a really great answer because of the things I just said, but also because it can actually save you time because you don't have to worry about cleaning your incubator as often. So just be sure to wipe up any spills so that microorganisms will be in contact with the surface. And there's no need to use chemical germicides since the copper itself is more effective and the copper is safe for cultured cells. In fact, those chemical disinfectants can be a problem if they're used on copper because as a natural element, the copper can react with many chemicals, resulting in a precipitate and affecting the copper surface. We should say that 100% pure copper will develop a black and green tarnish over time, as you can see on the pictures here. That tarnish can be surprising if you're not expecting it, but green is good. This is actually a good thing. The tarnish develops through a natural process called passivation, and that process results in the copper becoming even more effective. It even works faster, and the passivation strengthens and smooths the copper surface. So that's a lot of detail to answer your question, but my answer is there really aren't any downsides. Copper is naturally antimicrobial, safe for cells, and easy to maintain.
Well, that's some great information. And what I'm really realizing here is that there was a lot that I didn't know about all the different, choices for CO2 incubators. Are there any other features that we haven't talked about yet, that can help us control contamination?
Yeah, Megan, I love to talk about in-chamber HEPA filtration because there are a lot of misconceptions about this technology. HEPA filters are rated for their efficiency of capturing zero point 0.3 micrometer particles, which falls between the physical processes of impaction and interception, which capture larger particles, and diffusion, which captures smaller particles. As a result, in-chamber HEPA filtration does protect against viruses, mycoplasmas, and other microorganisms that could put your cells at rest. Our incubators filter the chamber air every 60 seconds and provide ISO Class 5 clean room quality air 5 minutes after a door opening.
I mean, that sounds great, Callie. I've heard HEPA filters are expensive and that sometimes microorganisms can still escape.
Yeah, I've heard those rumors too, Megan, and they may originate from manufacturers that can't offer in-chamber HEPA filtration because any HEPA filter requires a circulating fan to push that air through the filter. So if the incubator doesn't have a circulating fan, it just can't offer HEPA filtration. Most HEPA filters in a CO2 incubator last for 1 full year. They can be replaced in minutes, and they only cost between 50 and about 150 US dollars each. Callie mentioned the ISO Class 5 conditions. That's 10,000 times cleaner than normal indoor room air, so you can imagine how well that helps protect cultured cells.
Yeah, I mean, you did mention there's a lot of misconceptions out there, so thank you for debunking that rumor for me. There's so much to know about contamination control technologies. And I was actually thinking about another solution for this. Let me know what you guys think. What if we put a UV light in the incubator to control contamination? I mean, I think this sounds like a great idea if I do say so for myself.
Well, I actually get asked about this a lot, Megan, and we do have some history with this. Back in 1980, Thermo Fisher Scientific was the first to introduce a UV light into a CO2 incubator. We stopped offering it in 1998 because our customers told us that it was not effective. And fast forward to 2006, the American Biological Safety Association came out with a position paper that said a UV light is essentially ineffective in air with humidity above 70%. Since cell culture incubators generally provide 90 to 95% relative humidity, this explains why it doesn't work.
I think I'll stick to hosting Lab Talk and I won't try to come up with any new incubator solutions anytime soon. I'll leave that to the experts. So let's move on to sterilization techniques. Mary Kay, do you have a favorite?
Oh, yeah, Megan, I like pretty much everyone else. I like to keep things simple. I've had contaminated cultures, and I've had to clean and sterilize and disinfect CO2 incubators. So for me, as I said, I like it simple. And I think the easiest method of sterilizing is a high temperature cycle. You just remove the HEPA filter, drain the water, and push the button and leave it overnight.
I mean, that does sound simple. And I like simple too, Mary Kay, so I'm in for that. And it really does seem that most incubators offer a high temperature cycle now. So I have a question around that. Are they all pretty much the same? I mean, higher temperatures are better, right?
Oh, yeah, not necessarily. The US and European Union pharmacopoeias are careful to point out that it's not about just temperature, but it's the combination of temperature and exposure time that determines the efficacy of a cycle. Both of those pharmacopoeias state that you have to prove efficacy using prescribed heat-resistant microorganisms. Sterilization is defined by elimination of at least 1 million or 10 to the power of 6 of those specific microorganisms. So just saying an incubator has a cycle that heats to 180 or some other temperature, that doesn't mean very much. And most of those incubators with a 180-degree cycle don't actually reach 180 degrees in all areas. So microorganisms could survive that cycle in cold spots. As one example, I've recently seen a new white paper from a manufacturer that claims 12-log sterility assurance level, but those guys used vegetative, actively growing versions of those bacteria, and they're not heat resistant. So as always, any automated system should be tested and proven effective.
Yeah, I agree. I mean, proven sterilization certainly would provide peace of mind, especially since we already said that an incubator is a great place for microorganisms to live, as well as for tissue cells, right? So, one more topic, before we take some questions for the audience. I keep hearing about VHP or vaporized hydrogen peroxide. Is this safe and effective?
Megan, I really wish it were that simple. Hydrogen peroxide is a great disinfectant as it's effective against most microorganisms. And as consumers, we can buy it at our local stores in very low concentrations, around 3%, and it's pretty safe. But at high concentrations, it's a known carcinogen. Companies that offer VHP services for laboratories wear personal protective equipment and ensure that the vapor doesn't escape due to the dangers of the highly concentrated VHP. Working with highly concentrated VHP outside of this scope could potentially expose the lab, the staff, and equipment to this known carcinogen.
Okay, but I thought there were some CO2 incubator models that offer VHP, as a push button automated system, kind of similar to the high temperature sterilization cycles. Is that right?
Yep, you're right. There are some of these on the market today, and to be honest, we've heard quite a few complaints. It's really challenging to automate such a chemical approach because it's not always possible to remove and arrange the internal parts so that vapor makes contact with every surface. So that means that some microorganisms not exposed or not reached by that chemical could survive. But the automated systems do not neutralize that remaining chemical, and that can cause corrosion to the incubator components over time, and that puts cultured cells and people in the lab at risk. Once again, the bottom line is that any such system needs to be proven safe and proven effective. So for me, at least right now, I'd go with high temperature sterilization, as I said, or the guys in the suits who provide professional VHP as a service. Most of those services will provide evidence confirming the process was effective, the chemical is neutralized, and the air is now safe for humans and cell cultures.
With that, this wraps up our episode of Lab Talk for this week. I know I sure learned a lot today, and I hope everyone else did. Once again, wanted to thank our presenters, Mary Kay and Callie, for sharing their insights and expertise with us.